Hello everybody, my name is Aziz Tumeh. I am a PhD student from the fourth PGSB call. My project title is Structural Characterization of a Proapoptotic Part 4. This project is being conducted under the supervision of Professor Jörg Tabhan and Professor Hilal Zaid, and we are carrying this project at the Center of Structural System Biology, CSSB, outstation of Institute of Biological Information Processing IBI Special Structural Biochemistry Department, IBI-7. So, first, a small introduction about the Part 4 prostate apoptosis response factor 4. This is a proapoptotic protein with a tumor suppressor activity. It consists of 332 amino acids, and studies have shown that this protein is downregulated in a variety of cancers such as colon and prostate and renal cancers, while it's upregulated in neurodegenerative diseases such as Alzheimer's disease and Huntington and Parkinson's disease. What makes Part 4 a special protein is its ability to induce apoptosis in cancer cells, but leaving the normal cells or mortal cells are unaffected. So it's a good target to study and to know about this protein. Part 4 in apoptosis. So Part 4 has been involved in both extrinsic and the intrinsic pathway of apoptosis. So the extrinsic pathway is the most well-characterized mode of apoptosis by Part 4, and there is a little known about the intrinsic pathway. However, I will explain the extrinsic pathway. We have the protein, glucose-regulating protein 78, which is normally resident at the endoplasmic reticulum, and Part 4 is normally secreted outside of the cell, normal cell and cancer cells. So when there is an apoptotic stimuli, what will happen? Part 4 will bind to the glucose-regulating protein 78, and they both will move to the plasma membrane, where they will activate the fast-phasal pathway, apoptotic pathway. This dimeric, the fat protein will join them and they form trimeric. This will activate the procaspase 8, and then procaspase 8 will activate the caspase 3, and caspase 3 will cleave the Part 4 and to a segment we call it cleaved Part 4. And this cleaved Part 4, upon phosphorylation by protein kinase A, then it will be able to translocate to the nucleus and inhibit the nuclear factor kappa B and induce apoptosis. So this is the extrinsic pathway, which is the most well-characterized mode. However, Part 4 is also located inside the cell in the cytoplasm. So Part 4 can induce apoptosis, but in this case, it will not be translocated to the nucleus, but it will inhibit the atypical protein kinase C. So this enzyme normally, without Part 4, it will activate the nuclear factor kappa B, and the nuclear factor kappa B will promote cell survival. So the cancer cell will be survived. So Part 4, in case of Part 4, when it's active, Part 4 will inhibit the atypical protein kinase C, and once it inhibits this enzyme, then there is no activation for the nuclear factor kappa B, then the cell would go for apoptosis. There is a study was conducted in 2002 that P62 protein can join Part 4 and atypical protein kinase form at a ternary complex, and once they are formed at the ternary complex, Part 4 will not be able to inhibit atypical protein kinase C. So we want to use P62. In the next slides, I will tell that for the structure analysis. For the normal, the intrinsic pathway, which is about through the mitochondrial pathway, the Part 4 will induce the release of cytochrome C, and cytochrome from the mitochondria. Cytochrome C will activate the caspase 3 and the caspase 9, and the caspase 9 will activate the caspase 3, and caspase 3 will activate the Part 4. But as I said before, this pathway, there is no much details about the action of and the mechanism of Part 4 in the intrinsic pathway. So this cleaved Part 4 is my target in the PhD project. So Part 4 is cleaved, as I said, by caspase 3 at residuous aspartic acid 131 amino acid, generates two segments, a long one and a short one. The long one, we call it cleaved par 4, and it contains the SAG domain and the CC domain. And the cleaved par 4 has the ability to induce apoptosis in the cancer cell through its SAG domain. So par 4 domains. Par 4 has several domains that are conserved among human and mouse and rats. These domains are, we have here in the C terminal, we have the CC domain, and in terminal we have the SAG domain. The CC domain contains a leucine zipper domain, and this part is play a role in hetero and homo and hetero dimerization with other proteins in order to regulate the function of the bar 4, where the SAG domain, it's the minimum fragment that able to induce apoptosis in cancer cells, and it has a nuclear localization sequence domain within it, and in order to be translocated to the nucleus and induce apoptosis, it has the nuclear localization sequence. Also, there is a phosphorylation sites for in bar 4. There is a phosphorylation site for protein kinase A at the threonine 155, and there is also a phosphorylation site for protein kinase B at serine 249. So the C terminal, as I said before, this is the C terminal. So it ranges between 254 and 332, and this one was predicted to form a coiled structure. And this prediction was confirmed and solved by my colleague, postdoc, Dr. Uday Kumar. He has solved this structure of the C terminal, and he confirmed that there is a coiled structure with a leucine zipper domain in the part 4. And this structure was also confirmed by the CD spectral circular dichroism. As we can see here, there are two peaks at 222 and 208, which gives an typical coiled structure. And for a coiled structure, it consists of alpha helices, and these alpha helices are in heptad repeats, and heptad repeats is donated by A, B, C, D, E, F, G, and these that we have here in the C terminal, we have 11 repeats, and the first 5 repeats is responsible for the homodimerization domain, whereas the next 6 repeats is responsible for the leucine zipper domain, and they are responsible for the heterodimerization and binding with other proteins that will regulate the activity of par 4. So the N-terminal, this is the N-terminal, the SAG domain, it's ranges between 137 amino acid and 195, and it contains the nuclear localization sequence, and it's able to induce apoptosis, as I said, especially in cancer cells, but leave the normal and immortalized cells are unaffected. So, intrinsically disordered protein, or it's shortly known IDP, par 4 is considered and known as an intrinsically disordered protein. It means they lack a fixed three-dimensional structure. So, and we want to study the structure of this protein, and we want it to be in three-dimensional structure. Therefore, we need factors that can affect in the stability and the folding of the proteins, and there are several factors. Actually, we have the extreme conditions, means pH, low pH, and high concentration, because for the part 4, and because it's intrinsically disordered proteins, the repulsion forces between the amino acids is high. So we wanted to decrease this repulsion forces by at high salt concentration or pH, so that the net charge will be, the negative or positive charge will be decreased, so the repulsive forces will be decreased. Also, another thing, maybe the post-translation modifications, such as glycosylation, phosphorylation, and simulation. And BAR4, using the post-translation modification prediction tools that are available online, showed that BAR4 has a different site for these modifications, like glycosylation, phosphorylation, and simulation. And all of them can induce stability and folding of protein. Also, interaction with other proteins like ions like glucose-regulating protein, B62, and CRM1. Also, these proteins, when they interact with the BAR4, so the BAR4 can be folded, and we can get a 3D structure for this protein. So the aim of my research, so this part, the C-terminal part, was confirmed to form a coil structure with a leucine zipper domain, and as I said, it was done by my colleague, postdoc. My task now is to extend the structure outside of the CC domain. So till here, it's the structure is solved and analyzed, but the SAC domain, which is responsible for the apoptosis, is still needed to be analyzed. So my project will focus on the extended region outside of the CC domain to include the SAC domain. In this case, we call it cleaved BAR4. And in the methodology to do this, my research is divided into 4 stages. So we have the molecular biology, protein production, and biophysical characterization, and structural studies. In molecular biology, we want to create and have the required BAR4 clones. Therefore, I have 2 constructs for BAR4 to express in E. coli. We call it CCX short, means CC domain, and CCX long. This one consists of 197 amino acids, and the CCX long 280 amino acids. And these constructs to be expressed in E. coli, Escherichia coli, and also we have 2 constructs to be expressed in insect cells, and also the same, nearly the same construct, CCX short and BAR4 full length. So for protein, BAR4 protein expression in E. coli, we use this vector. We use for the E. coli BET11A, and this is the clone for the BAR4. We transformed it in the E. coli RIL cells, and then when the clones, we can see the clone, then we do inoculation to the main culture, and after that, the inoculation, we put it in the shaking incubator for at 37 degrees for 120 RPM. And when the density OD600 reached from 0.6 to 0.8, we add IBTG, and this IBTG is a protein induction in order for the cell to produce the protein. And here the protein is produced. Then we harvest the cells by centrifuge, and then we have the cell pellet. For purification, our protein, we put a strip tag, which is, let's say, like a mark for the protein, and we use a streptactin resin, and this resin, so it has to be equilibrated with the equilibration buffer. Then we add the cell lysate, and the cell lysate will contain the target protein and other proteins. So the other proteins will be in the flow through, and the target protein will be bound to streptactin resin. Then we do the washing step, and in the washing step, we will remove any protein that are not bound to the resin. Then after that, we elute the protein we have different methods to elute the protein. We usually use maybe decreasing the pH, or we also use disthiobiotin also as competitive to bind to the streptactin resin. So now I'm facing a problem here with the BAR4 that is eluted is contaminated with the DNA, and we want protein BAR4 to be clean of DNA. We want to get rid of this DNA, because if there is a DNA bind to my protein, then this will affect in the interaction with other proteins, and I want to understand the structure of the protein with binding it with other proteins. So if there is a DNA, this will affect in binding the target protein, which is part 4, to the other partners proteins. Thank you. But before, I would like to thank the PGSB for giving me this opportunity to be a PhD student in the fourth goal, and also I would like to thank my supervisors, Palestinian supervisor and German supervisors, and thank you.